And Leela, we are doing the first your presentation, then interview, then question answer.
Okay.
Okay. Generally, we are doing the question answer after the just finish the webinar.
Okay.
A lot of times I used to get very nervous speaking to overseas clients.
Okay, we are live now.
Okay. So should I start my slides already?
No, actually Liberty will ask you to do that.
Okay.
If you want to check the slides, okay, you can do that. Or no problem, you can start whenever Liberty asks you.
Okay.
No problem. So Shoma, I'll start.
Sure.
You unmute yourself. Welcome everyone to our 33 webinar in this series. This is another international webinar. It's open for all and it's free for all. Our intention to promote plant science internationally, and we are trying to build a community and a platform for plant science research and plant loving people. I am very fortunate. We have today we have a very dynamic and young researchers. Dr. Leela. Liberty, my friend from UC Davis, will introduce her. Shoma will Shoma will interview, and today we have another guest, Dr. Sujan Sir, who will assist me for the YouTube questions collections. I request all the participants, you already have the application feedback certificate application link to your email address. If you don't have, please check your spam folder. If you're not getting the link, don't worry, you email me, I will send the link once again. But this link is dedicated for those people, those are registered for this webinar, but it's free for all. Anyone can watch it from our YouTube channel. Any PPT or PDF will not available for the participants, but the recording of this webinar is free and open, and it can be viewed many times from our channel. It's my request to subscribe to our channel to keep more updated on plant science research internationally. And if you need any information, go for the bioingene.com. It's our website, and we all have a different social media. You can contact us in different ways. I request Liberty to introduce Bioingene and our speakers.
Thanks, Joel. It's great to be here. Welcome everybody to the Bioingene webinar series. The webinar series is a platform from which plant scientists can present their research to the world and future scientists can gain knowledge, perspective, and inspiration. Today is our 33rd webinar of the series. We welcome all the viewers who have joined us from around the world. If you are new to Bioingene, please note that after attending today's talk, you can apply for a certificate of participation. Submission of the feedback form is mandatory to receive a certificate. We have already emailed the feedback form certificate application link to the registered participants. The password will be provided multiple times in the YouTube chat box during the question and answer and interview session. This comes after the presentation. When you fill out the feedback form, please mention the full name of your institute and the full institute address. You can collect your participation certificates after 2 days from our website. Bioingene does not send certificates through the email. Please visit our website to register for upcoming webinars. You may submit articles in our Bioingene Plant Sciences Journal. This is a peer-reviewed, open access international journal. You may also send your posters to be displayed on our website. Please connect with Bioingene on Facebook, YouTube, Twitter, and LinkedIn. Now, the topic for today's webinar is 200 Years of American Cranberry Domestication and Status of Fruit Rot Research by Dr. Leela, Sai Sree, Uppala, Cranberry Pathology, University of Massachusetts, USA. Leela grew up in a rural part of southern India. She received her B.S. degree in agriculture and M.S. degree in plant pathology. She received her Ph.D. in plant pathology from Auburn University in Alabama United States. Before joining the University of Massachusetts, she conducted research on diseases of leafy vegetables in India. Peanuts at Auburn University, Alabama in the United States, Kentucky bluegrass and apples at Oregon State University in Oregon in the United States, and rice, which we all know and love, at Texas A&M University in Texas. Leela's research efforts at UMASS Cranberry Station focus on the development of novel, environmentally friendly, cost-effective, and sustainable integrated disease management strategies for organic and conventional cranberry production systems. Other areas of her research include cranberry microbiome, disease forecasting, and fungicide resistance. Leela's extension efforts focus on 1, establishing UMass Cranberry Diagnostic Lab and providing timely identification and management guidelines to growers. 2, educating growers, crop consultants through extension and outreach. And we are highly honored to have her on our platform. I now request Dr. Leela to please share her screen and start her talk whenever she is ready.
Thank you so much, Liberty, for your introduction. I really appreciate it. And I'm extremely honored to be part of this webinar series. I'm looking forward to this presentation. Can you all see my shared screen, right? Yes. So as Liberty mentioned, I'll be talking about 200 years of cranberry domestication and current status of fruit rot research. So the outline of my presentation is introduction and some fun facts about cranberry and the history of domestication and what we know about cranberry fruit rot so far and what kind of research is being conducted in my program in Massachusetts. So once again, I'm introducing myself. Liberty already did a wonderful job. I'm from southern part of India, from Andhra Pradesh, from a village called Korumamidi, where our village is agriculture based, and most of the villagers depend on agriculture for their livelihood. We grow rice, coconut, banana, a lot of vegetables and sugarcane, and of course, cattle for milk. And as Liberty mentioned already, I did my undergrad from Acharya N.G. Ranga Agricultural University in Andhra Pradesh and master's from Kerala Agricultural University through ICR fellowship, thanks to ICR, and Ph.D. from Auburn University in Alabama. And I got opportunities to do postdoctoral research and extension training from Oregon State University and Texas A&M University. Now, at present, I'm an extension assistant professor, currently working at the University of Massachusetts Amherst Cranberry Station. So the main campus is University of Massachusetts, located in Amherst, and the Cranberry Station is located 140 miles from the main university campus. We are located in East Wareham. Ours is an extension and research and outreach center. Our goal is to serve the Massachusetts cranberry industry and cranberry growers, who are mostly located in southeastern parts of Massachusetts. That is why we have our station in southeastern part of Massachusetts. This is a beautiful aerial view of our station. So as you see, here are the buildings and greenhouse, and this is a cranberry field. And in Massachusetts, cranberry fields are called bogs, B-O-G. So cranberry bog, we have 11-acre bog on site. So let's talk about some fun facts and history of domestication. Cranberries were not always cultivated. Native Americans, they used to gather wild berries and use them for many forms like medicine, food preservatives, and as dyes. And they also used cranberries as poultice for wounds when mixed with cornmeal, and it served as excellent cure for blood poisoning. If you are wondering what is poultice means, it is a soft, moist mass of material, typically of plant or flower, applied to body to relieve soreness and inflammation, and it is kept in place with a cloth. So these are some of the historical uses of cranberries, and Native Americans also used cranberries in tea to calm nerves, and they used cranberries as dyes to brighten the blankets and rugs. And they used to make pemmican, which is nothing but a mixture of deer meat or fish and mixed with berries, cranberries, and pounded into pulp and shaped into a cake and dried it in the sun. Native Americans were the first to make sweetened sauce using maple sugar. Cranberries by itself are tart or sour in taste. So they used to mix maple sugar and make a sweetened sauce. Now in the US, cranberry sauce is so significant, especially for Thanksgiving festival. It is almost like, you cannot imagine Holi without colors, right? Similarly, you cannot imagine Thanksgiving festival without cranberry sauce. It became so part of Thanksgiving festival now. And it is known to have benzoic acid. So the cranberry is known as, that is used as food preservative in many foods these days. Of course, cranberries were not always called cranberries, as it is a Native American fruit and first used by Native Americans. Different tribes of Native Americans called it by different names. For example, Narragansett tribe called it as sesamaniash, and South Jersey tribes called cranberries as ibeme, and Algonquin tribe called it as atoqua. When the Algonquin tribe shared these fruits with European settlers, the first settlers who came from England to the US to settle in the United States, the current day United States, they did not find the English equivalent for cranberry, and they started calling it cranberry because of the flower's resemblance to the head and bill of a European crane. If you see, the flower has this shape, which is almost same as the head and bill of a European crane. So that they started calling it cranberry, which over time got shortened to cranberry. And of course, I'm using a lot of terms, which I really hope I can explain before we move to the next part of presentation. So we all know that Christopher Columbus came to the current US like in 1492 and then discovered this new land. So after that, Europeans started colonizing Americas. So before that, only Native Americans, the indigenous people, were living in Americas, North America. And then the European settlers who first traveled from England to North America were called pilgrims. They traveled by a ship called Mayflower. The first ship that brought these pilgrims had around 140 people. So that's called Mayflower. And then they first came to Plymouth area in Massachusetts, and then they started settling in surrounding areas, and slowly they moved across the United States. The first settled area, the six states are called New England. That's called New England now. And I'll talk about Thanksgiving in the next slides as well. Once again, Native Americans are also known as American Indians, First Americans, or Indigenous Americans. They are the indigenous people of the United States who have been living in the current US since 15,000 BC. And there are 574 federally recognized tribes currently living in the US. They are notable for a wide variety and diversity of lifestyles, languages, clothes, customs, and beliefs. So the pilgrims, as I mentioned, are the English settlers who came to North America on a ship called Mayflower in 1620. And they established a colony called Plymouth Colony, and which is in Plymouth, Massachusetts. If you see the map here, the blue United States map here, Plymouth is in the northeastern tip of United States right here. And they migrated to Plymouth after 10 weeks of harsh weather and facing harsh weather on the sea, to flee the religious persecution that was happening in England. They wanted to get that freedom and wanted to practice what they believe in, and then fled to the newfound land, or the United States, current day United States. And as I mentioned before, from Plymouth, they started spreading to nearby areas first, and 6-state region in northeastern corner of the United States is called New England. It is one of the oldest settled parts of the United States. And slowly, of course, they started moving across the country, then they all the way up to Oregon or Washington, the Pacific coast as well. And let's talk about Thanksgiving. Thanksgiving, current day Thanksgiving, is a national holiday. It's celebrated across the country, and it's a day of giving thanks and sacrifice for the blessing of harvest of the previous year. And it's usually celebrated on the second Monday of October in Canada, and on the fourth Thursday of November in the United States. That is current day Thanksgiving. So what about the first Thanksgiving? When did the tradition start actually? So when the Pilgrims, the early European settlers, when they moved from England to the newfound land, that is Plymouth, Massachusetts, to Mayflower, it was not an easy journey for them. Not on the sea, not after reaching the Plymouth, because they had several harsh encounters between them and the native indigenous people. And after all that said and done, they did cultivate crop and they survived here. So at the end of their first year or first season, they wanted to celebrate their harvest in the new world. The new world is like a place where they entered. And in around October 1621, they wanted to celebrate it with first Thanksgiving. That is how the tradition started. The feast lasted for 3 days and was attended by over 19 Native Americans and 53 pilgrims. And here is a paint I really liked from the internet. It shows the Thanksgiving feast attended by the Native Americans and pilgrims. And if you check the first Thanksgiving feast, there are various components like cod, bass, venison, ducks, geese, and lots of fruit tarts like strawberries and grapes. But we don't see cranberry here. So why are we talking about Thanksgiving in the midst of cranberry presentation, right? Because cranberries are so symbolic of current Thanksgiving festival, but may not be the first Thanksgiving festival. And the cranberry, even if it made any appearance at all during the first Thanksgiving meal, it was as a minor ingredient in stuffing of roasted birds. And cranberry became part of a Thanksgiving meal after sugar was introduced to New England. That is when they started making this sweetened sauce, which became part of current Thanksgiving. That's why we're talking about Thanksgiving. So let's talk about cranberry domestication. The first actual cultivation of the cranberry was attributed to Henry Hall, a Revolutionary War veteran who lived in North Dennis, Massachusetts on Cape Cod, which is not that far from where I live right now or where our cranberry station is located right now, which is only a couple, maybe 20 minutes from where we are. That is where the first actual domestication started. As I mentioned in previous slides, Native Americans only used wild berries, but then cranberry domestication started in around 1816 when Mr. Henry Hall transplanted some of the vines into a fenced area to protect them from his cattle. And around 1816, he started, and by 1820, he started sharing the harvest with his friends and family in Boston and New York City. Soon, the word spread rapidly and many individuals started transplanting sorts of cranberry vines for their own yards in Cape Cod and throughout Massachusetts. Today, Massachusetts is the second largest producer of cranberry in the world. And if you see cranberry, before I came to Massachusetts for my job interview, I always used to imagine cranberries as bushes, but they are not. They are literally perennial vine trailing on the ground. If you see, this is a cranberry field paint. So it is literally on the ground. That is where they're harvesting cranberries. And later, it spread across other regions as well from Massachusetts. But cranberry is the official state berry, and cranberry color is the official state color of Massachusetts. And cranberry is the official drink of Massachusetts. Two principal cultivars that are still grown in Massachusetts, like Early Blacks and House, were selected from Harwich and Dennis area of Massachusetts, respectively. And in and around 1840s, to this date, we still have these cultivars here. And around 1860, when Edward Sackett of Sackett Harbor from New York traveled to Berlin in Wisconsin to inspect some land, and he observed around 700 acres of wild cranberry vines there. And then he decided to cultivate the marshes. See, there's a new term here because previously I talked about bogs, because in Massachusetts, we call them we call cranberry fields as bogs. But in Wisconsin, we call the cranberry fields as marshes. So Edward Sackett, in 1860, when he found 700 acres of wild vines, he sold his cranberries in Chicago for as high as $15 per barrel back in 1860. That's big. And today, Wisconsin is the top producer of cranberries in the world. And it is not only their official state fruit as well, but also their number one crop. And it was also recorded that cranberries were cultivated in New Jersey by 1835. So 1820, Massachusetts, Henry Hall, and 1860, there was record of wild cranberries in Wisconsin, and 1835 in New Jersey, there were records. But later, the cranberries were introduced to other regions in the United States, like Oregon and Washington, and even to other countries like Canada, Chile, and some countries in Europe. Initially, Boston, in Massachusetts, became the major marketing center for cranberries. That is where the shipping happened from United States and other regions of the United States and Europe. And barrel became the standard unique measure of production of cranberry. One barrel is equal to 100 pounds. And sailors used cranberries as a source of vitamin C to prevent deficiencies, especially scurvy. So cranberry is a rich source of vitamin C. Other fun facts are, during the Second World War, American troops consumed almost 500 kilograms of dried cranberries every single year to prevent scurvy. And then slowly the industry started expanding, especially when Reverend Benjamin Eastwood published his book on cranberry cultivation in 1860s, and prices were so high due to high demand during the Civil War. And the industry also expanded with the coming of railroad, transportation made easy. And the second important publication was on cranberry culture by J.J. White in 1870. That also helped in expansion of cranberry acreage. By 1900, which is around 80 years after first domestication reports, around 21,500 acres were in production. And around 1930, industry reached its highest with 27,640 acres harvested. But of course, just like any other life events, there will always be challenges and setbacks. And in around 1930s, due to the Great Economic Depression and the onset of false blossom disease, which is a phytoplasma disease, acreage reduced. And in 59, there is a Secretary of US Department of Health and Education and Welfare. He published a book or a report on aminotriazole amounts in cranberry. In certain cranberries, there was aminotriazole herbicide, which is a suspected carcinogen. When he reported that, and it scared people, and then that also resulted in decreased acreage or decreased demand for cranberry. However, the industry again rebounded in late 1960s, and then it went all the way up to 40,800 acres by 2018. In the last 2 decades, from the 40,800 acres is the United States acreage. In the last 2 decades, it even expanded to other countries, as I mentioned before, like Canada, Quebec, and Chile. And once again, the major US growing regions are Massachusetts, Wisconsin, New Jersey, Washington, Oregon, and some other states like Michigan, Connecticut, Rhode Island. And if we put together all the cranberries that are grown in North America side by side, you could actually connect Boston, which is in northeast part of United States, to Los Angeles, which is in Pacific coast, more than 565 times. Think about that, that's big. And Americans consume almost 200 million kilograms of cranberries every single year, including 40 million just during Thanksgiving. And cranberries that are grown here are sent around the world, and top 10 export markets are Europe, United Kingdom, Mexico, China, Canada, Australia, Chile, Taiwan, Israel, and South Korea. And US acreage as of 2018 is 40,800, and world acreage as of 2018 is 61,000. Massachusetts has 4 major receiving stations. The receiving stations are nothing but processing industry. So they receive the fruit and they sell as fresh fruit, which is very small percentage, and process the remaining fruit into various value-added products. They also have 2 major manufacturing plants and 2 corporate headquarters. It's really a unique crop and unique cultural practices. Let's take a peek. So it is also called American large-fruited cranberry. Scientific name is Vaccinium microcarpum, and it prefers acidic soils that are moist, well-drained, and high in organic matter. Most of the cranberry soils are more than 90% sand, and major yield components are number of fruits per upright. Upright is not the vegetative stem of the cranberry, and number of flowering uprights per unit area. These are the yield components. They have high impact. And fruit quality parameters of commercial interest include fruit size, fruit color, and fruit firmness. So again, it is a trailing vine, that perennial crop that grows on the ground. It's not a bushy plant. As you see, I sat on the bog. So when I first joined here, I was like, where are the walkways? For all other crops, you would find walkways where you walk and then where the plants are. But as it is a trailing vine, it would be hard to separate walkways. So you just walk on the bog, and then cranberries are really, really resilient, amazing crop. And if you take a closer look, this is how the plant is. These are called runners. They run on the ground, and with vertical stems, which are also called uprights. These vertical stems produce flowers, which become fruit, and they have fine fibrous root system. Perennial vine, usually we recommend renovating the bog once in 30 years, but there are several bogs which are more than 100 years old and still producing pine in Massachusetts. So it's a perennial crop, and as I said, uprights produce these fruits. There are more than 70 varieties of cranberries available, and there are natives like early blacks and halves, which produce relatively smaller fruit and compared to the first generation hybrids and super hybrids, which were developed later on. And it is one of the 3 native fruits that are cultivated in North American continent, like blueberry, Concord grape, and cranberries. These are the only 3 native fruits that are cultivated in North America. As I mentioned again, it is a unique crop that occupies unique environment like acidic soils, low nitrogen requirement, and sand is more than 90%. So most agronomically important crops would not survive under the conditions which are optimum for cranberry. Here are some fun pictures shared by our research station director, Dr. Hilary Sandler. This is traditional planting. This is vegetative propagation, right? They plant these vines or plant with plugs with machines like this. Another unique practice is sanding. The war veteran who first domesticated cranberries in Massachusetts, Mr. Henry Hall, he observed that some of his best producing cranberries are near his home that were receiving regular dune sand driven by northeastern winds. When there was more sand, they are producing more. Then it became a practice of sanding once in every 3 to 4 years. That's what is recommended, to sand 0.5 to 0.75 inches. Yeah, 0.5 to 0.75 inches every 3 to 4 years. Sanding is known to stimulate new vine growth and suppress insects or pathogen inoculum and improve drainage, and also help the breakdown of the trash layer from any plant tissues or like leaves or uprights that are falling on the bog. The sanding helps break down of the trash layer on the bog and make the nutrients available to the plants. And sanding can be applied directly to the vines in the spring or fall, or in winter, when there is ice on the bog, they can apply sand, which would later fall on the plants after ice melts, or by barge sanding, which is nothing but sanding when there is flooding. So I will explain cranberry flooding later on. It is one of the unique practices again. So most growers use specialized sanders that many have built themselves. Here you see, it is sanding directly on the vines or sanding on the ice in winter. And barge sanding barge sanding. It is in flooded cranberry fields. If you see this, you don't think it's a field, right? But it is actually cranberry field. So cranberry, which evolved in wetland habits, so it can tolerate periodic long flooding. So in the northeastern area, around December 15th or mid-December to mid-March, growers just flood their whole cranberry fields and let the water sit. So that is the way of protecting cranberry plants from dry, cold winter weather. So they just flood the whole field for almost 3 months and let that water stay like that. So this is a cranberry field. So and also the machine is just going on top of the plants, if you see, and it is leaving the sand into the field. So That's why I've been saying it's a resilient crop. Machines grow on top of them, people walk on them, but it still survives, and years and years, like hundreds of years. So another other practices are pruning and mowing, as it is trailing vine, perennial vine. It produces runners and more uprights every single year. So as time goes, you have take growth. So you have to prune them to remove excess runners. When the runners are in excess, upright stand become dense, and that limits the light penetration into the canopy and thereby decreases yields, because it decreases flower bud initiation, and also it limits the movement of pollinators into different flowers. Cranberry depends on cross-pollination from honeybees and bumblebees. So if there is a thick canopy, it won't let the pollinators to reach all flowers. So pruning facilitates the use of dry harvesting equipment. So yeah, I'll talk about harvest too. So here is a pruning. They are removing excess runners by pruning equipment. And mowing is little, again, it's also another method of pruning, but a little heavy. Other unique cultural practice is late water. I talked about how flooding is used as a means to protect cranberry plants during winter, right? That flooding happens from mid-December to mid-March. However, in around 1940s and 50s, in New Jersey and in Massachusetts, researchers observed that when the farmers left the flooding beyond March 15th, they used to observe more benefits, like beneficial effects on reduction of insects, pathogens, etc. So they thought that late water using winter flood beyond March 15th might have some benefits. Then this became a cultural practice called late water, especially in bogs that are old and not so healthy. For them, late water used to be even more beneficial. That is when they realized that some bogs may benefit from this late water practice. Now it is recommended once in 3 years. Sanding is recommended once in 3 to 4 years. Pruning is recommended once in 3 to 4 years, and late water is holding again in spring flooding for 4 more weeks from mid-April to mid-May. So that is also recommended once in 3 years. Again, as I said, it's a 30-day spring reflood. I'm using the word reflood because so in December 15th to March 15th or mid-March, winter flood, and then take that out, and then again reflood by mid-April, and then leave it till mid-May. That's called late water. It's a spring reflood practice several weeks after the winter flood has been removed and before the plants have lost dormancy. Cranberry plants go into dormancy right after harvest around October, end of October to November, they start going into dormancy. So this late water should be applied after the winter flood is removed and before the plant lost dormancy. Dormancy symptom, or that plants are still in dormancy. If you see the buds are still red and tight, the bud should not open. Before that, you should apply late water, which is once in 3 years. It is known to suppress insect, fruits, weeds, and improve keeping quality of the fruit. Harvest. This is other fun cultural practice or practice in cranberry, which is so unique to cranberry. So olden days, they used to harvest cranberries by dry wooden scoops, so which is really laborious. You have to sit down or bend and do it. It is a lot of work, as here it is me, I was doing with this wooden scoop. There are different kinds of wooden scoops or different sizes of wooden scoops available. It is used for harvest until 1940s. So you just move the scoop in the direction of the vine and all the berries come into the scoop. That is one method. But later on, mechanical harvesting machines were developed. This is called dry picking because there is no water involved. You just move the mechanical harvesting machines on the bog that takes the fresh fruit, and these are the mechanical harvesting machines. As you see, they put them in these bags, which are moved to the bins you see on the bog. And then once the bin is full, the helicopter would come and lift it up and move to the ground. And that would be transported to the receiving stations. This is called mechanical dry harvesting method. And the next method, which is the most common methods of harvesting nowadays, is water harvesting or wet harvesting. This is mostly used for processed fruit, or this is mostly used for fruit that is destined for processed market, not fresh market. So these are called egg beaters. These machines, so for this wet harvesting method, fields are flooded, cranberry fields or cranberry bogs are flooded with 18 inches of water, and these machines would go into the bog and they'll start releasing the berries from the vine. You know, they beat the canopy and the berries would get released into the water. And because of the air vacuum in the cranberry, they would float on top of water. They won't sink, but they would float on top of water. And whatever, all the berries that are floating on top of water here would be corralled to one edge of the bog. See, if you see in the previous picture, berries are everywhere in the bog. But here, all the berries were brought into one area. So they will be corralled and brought to one area of the bog, and they will be sucked into the truck and then transported to the processing plant or the receiving stations. Here you see the berries are being harvested or being collected into the back of the truck. And then they will be moved to the processing industry or the receiving station. That is where they will be processed into various products. If you see, this is the receiving station, all the berries are being moved and they will be processed into various products. Could be juice or concentrate or several other products like sweet and dried cranberries, powders, etc. Or even sauce, as I mentioned before. And fresh fruit, for fresh fruit market, which is relatively small, it is only 3.6% of the total Massachusetts crop is used as fresh fruit or is sold as fresh fruit. And for fresh fruit market, berries are dry harvested. And again, this is evolving. The processed cranberry products are evolving day by day. More and more products are being added to the counters. There are several products nowadays. And health properties of cranberries. Cranberries are considered to be superfoods due to high nutrient and antioxidant content. And research has linked cranberries to a lower risk of urinary tract infection, the prevention of certain types of cancer, and even improvisation of or improved immune function and decreased blood pressure. That is why cranberry is considered as superfood. Now the most fun part, cranberry diseases. So as a pathologist, what else can interest me as much as diseases, right? So for any particular disease to happen, as we all know, environment is important, like the favorable environment, susceptible host, and virulent pathogen are needed. That is when disease occurs. In cranberries, a spectrum of diseases which are very unique are there. And most of the diseases in cranberry are caused by fungal pathogens. Cranberry fruit rot is a major fungal pathogen disease. And there are other diseases as well, like upright dieback, Phytophthora root rot. but fruit rot is a major disease which is consistently prevalent from one season to the other. The first fruit rot was observed just 5 years into domestication. But the first research began during the early part of 1900s when Dr. C.L. Sree and co-workers from USDA started detailing the fungi that cause diseases in cranberries in Massachusetts and New Jersey. That was in early 1900s in Massachusetts and New Jersey. But in Pacific Coast, in Washington and Oregon states, cranberry disease research only started around 1922. And USDA sent Henry Bain to Pacific Coast for 4 consecutive summers to study diseases in Oregon. And relatively few diseases are caused by bacteria or viruses. As I mentioned, fungal diseases are more prevalent and more common in cranberries. But there is an exception, of course. It is false blossom disease, which is caused by a phytoplasma. And it was initially considered caused by virus, but later they realized it's a phytoplasma disease. It almost wiped out cranberry culture in New Jersey and in Massachusetts in early 1900s, like 1920s and 30s. But then later they discovered that this disease is vectored by blunt-nosed leafhoppers. When the cause of the spread is discovered, then they started controlling these vectors. That is when this disease subsided and cranberry cultivation continued. And if you see the distribution of economically important diseases in North America, Northeast, especially New Jersey and Massachusetts, have more diseases than any other regions because of hot and humid summers we have here, which are congenial for fungal populations. So major diseases are fruit rot, root rot, fairy ring, upright dieback. Among these 4, fruit rot is more prevalent from one season to the other. Fruit rot is a disease complex. It is not caused by one single pathogen. If you think about rice sheath blight, we all know it's caused by Rhizoctonia solani. Same thing, with different diseases, we know these are caused by this one particular pathogen. But on the other hand, cranberry fruit rot is a disease complex. It is associated with over a dozen fungi, and it is divided into fruit field rot and storage rot. If you observe symptoms during the field before harvest or around harvest, it's called field rot. And then if the symptoms of rot appear after harvest in storage, they're called storage rot. Fruit rot is very important because of the restrictions or regulations associated with it. Fruit deliveries beyond 12% rot, like if there is a truckload of cranberries, a farmer is delivering it to a processing industry at the receiving station, they check the number of berries and rot, or they take a sample and observe the percent fruit rot, and if the delivery has more than 12% of rot, the load is heavily penalized. If the deliveries are greater than 20% rot, they may just reject the entire load. That means a farmer spent the whole cropping season producing fruit, and then if they cannot control fruit rot to the level of less than 20%, they might just lose everything. So that's why it is even more important to control fruit rot. So as I said, since 1922, several researchers reported various fungi associated with fruit rot, and it's a complex caused by many different fungi. In some regions, some fungi are more prevalent than other fungi, and these also varies. What fungi appears in what levels, that also varies from one season to the other. So there are many, many unknowns about cranberry fruit rot, even 100 years after the first initiation of cranberry fruit rot research. Here are the various scientists, who discovered different fungi associated with fruit rot. So here are the different fungi and the spores, spores of various fruit rot fungi, like Allantophomopsis, Coleophoma, Coleotrichum, Epicoccum, Fusicoccum, Pestalotia, Phycelospora, Phomopsis, Philosticta. These are the pathogens we commonly observe in Massachusetts. And of course, for a disease to happen, we also need susceptible host, which depends on various factors. Not all are fully understood, but in general, we all know that canopy density, how well the air is circulating in the canopy, and the health of the vine, and the level of resistance, they all play important role in disease occurrence. So we don't have completely resistant cultivar for cranberry fruit rot, but we do have some which are more resistant than others. And favorable environment is also needed for disease occurrence for any particular disease, including fruit rot. And again, most of these factors are not well understood yet, but we know that weather parameters and local microclimatic factors are known to have great influence on fruit rot incidence. So let's see what management strategies we have right now. So of course, for any integrated disease management, we have three components: cultural control, chemical control, and biological control, right? And cultural control is any method that reduces the fruit rot inoculum and manages the canopy in such a way that air circulates well and canopy density is not so high, and that improves that causes congenial conditions for fruit rot pathogens are all important cultural management strategies. And fungicides for chemical control. Currently, we don't have any data on biological control methods for cranberry yet, but I'm working on that. Hopefully, we'll have some data in the near future. So again, cultural control and chemical options are there, but nowadays, more and more cranberry fields are cultivated with new generation hybrids. Not the native cultivars. Native cultivars are known to be little more resistant to fruit rot compared to the new generation hybrids. However, for the new generation hybrids, as you know that they are more susceptible, more farmers started using chemical control options compared to the cultural methods. So slowly, there was definitely over-reliance on chemical control compared to cultural methods when the chemical control options were available. So again, the percentage planted varieties changed over a period of time, and natives started reducing and hybrids started increasing. And then when the hybrids are more cultivated and there is more fruit rot and chemical control options are available, or farmers started using chemical control methods. And natives are smaller fruit. On the other hand, hybrids are bigger. So the yield is high. You cannot blame farmers for changing to, although susceptible, but high yielding hybrids. So before we talk about the fungicides that are available and that our farmers are using, and other aspects of chemical control, I would like to define FRAC grouping because I'll be using this term a lot. So the Fungicide Resistance Action Committee develops code numbers and letters for every fungicide to distinguish the different groups of fungicides based on the mode of action. So different chemicals were available since 1967. Here are some cool old disease control charts developed by the University of Massachusetts Cranberry Station. I found from 1967. There are several fungicides here. I don't expect you to all see these small letters, but just wanted to share these cool charts. So they have various compounds there like Maneb, Firbom, and other compounds back in 1967 itself. This is a 1989 chart, and there are several groups of fungicides available then. And again, chlorothalonils and other coppers, and there several were available since then. But now, fast forward to 2018, we don't have many available because some of the groups of fungicides we lost to revised maximum residue limits. There were several revisions to the MRLs, and we only have these groups of fungicides available right now. So among these, chlorothalonils and mancozebs are the go-to fungicides for farmers. They like these two because they're broad spectrum, highly effective compared to group 3 and group 11, which are site-specific newer generation fungicides. Coppers and polyaxin zinc salt, we don't have so much data and not many growers believe in them. So thus, we only had chlorothalonils and mancozebs and these other three. I use the word had because starting from 2018, there were some changes to MRLs of these compounds as well. So for the past 2 decades, more than 90% of cranberry acreage received chlorothalonil fungicides. You can imagine how important this tool is for fruit rot management. And more than 27% cranberry acreage received mancozebs. But in 2019, the export markets suggested that they are lowering the maximum residue limits to 0.01 parts per million from 5 parts per million. And in April 2020, we also received similar notification to change the MRLs of mancozebs. So again, chlorothalonils and mancozebs are the most commonly used fungicides, broad spectrum, and their MRLs are being revised. That left us with group 3 and group 11. That's all. These two are the groups we have available. They're site specific and prone for resistance if we use them again and again. So, and there is definitely no one size fits all approach in fruit rot management. So it depends on what is the prior year fruit rot incidence and whether you are cultivating susceptible varieties, relatively susceptible varieties, or whether you are using it on new crop, newly established vines, or several years or decades old crop. So you have to understand how many fungicides you are going to use. And some people use 2, some people use 3, some people use 4 to 5 applications, depending on the need for their particular bog. Once again, if you go back to this slide, we only have group 3 and group 11 fungicides. And how are we going to use 4 to 5 diversified fungicide program? So the chances of resistance are significantly high if we rely on these chemicals again and again. So thus, it brings us to my research program. So this is current status of fruit rot research. My short-term and immediate goal has been, for the past 2 years since I started, has been to preserve the effectiveness of the effective fungicides. We only have 2 groups. We need to protect them because if they develop resistance, fruit rot fungi develop resistance to these 2 available fungicides, we'll be in trouble, So that's why I've been advocating preserving the resistance monitoring and preserving the durability of these effective fungicides by diversifying as much as we can, even with coppers or whatever the biologicals that are available in the market, although we don't have much data yet. But simultaneously, I have been also evaluating fungicides with different modes of action from different FRAC groups. So for this, I initiated some studies like multi-state evaluation of chlorothalonil and mancozeb alternative fungicide regimes for their effects on fruit rot and fruit quality, because chlorothalonils and mancozebs will no longer be available at the level we had before. So we started this multi-state evaluation in New Jersey, Wisconsin, and Massachusetts, and we're evaluating novel fungicides and also Organic Material Review Institute approved products, because we also want to have biologicals in our toolbox, not completely conventional fungicides. And also, I have been advocating cultural practices. There was some gap in implementation of cultural practices because of the availability of effective fungicides for the past several decades. But now there is a need for cultural practices again. And there is a resurgence in interest in farmers as well. So here are several groups of new fungicides I have been evaluating for the past 2 years. So other than group 3 and 11, as you can see, we have 2, 7, 9, 12, M1, and biological control methods. I have been evaluating all these with a goal to expand the toolbox and diversify it. In 2020, I evaluated several compounds, and some of them showed extremely high efficacy in reducing fruit rot. Some are little still better than non-sprayed control, but little less efficacy than others. So we are making progress in evaluating novel fungicides and identifying them. So we, as I said, we identified 6 treatments from novel groups of 7, PN7, and coppers, and some biologicals that reduced fruit rot significantly. And we also found other treatments that reduced significantly compared to non-sprayed control, but not as good as the 6 treatments I mentioned above. So we are making progress in that direction. And some of these fungicides also had yield advantage compared to the non-sprayed control, but some other compounds which are effective didn't have no significant differences from non-sprayed control, but they had a good effect in reducing fruit rot. So, and these compounds also resulted in optimum fruit quality, like weight per berry, firmness, and so total anthocyanin content. They all had optimum levels of fruit quality, which is a good news because, you know, we want compounds that can diversify our toolbox, that can effectively reduce fruit rot while enhancing or maintaining the good fruit quality. And other research area I started is identifying critical criteria to develop a decision-making model for the implementation of late water floods in cranberry production. I talked about late water a little bit before, right? So late water is a spring reflood practice which happens from April 15th to around May 15th, 1 month spring reflood, where they flood the bog again and leave the water for a month in spring, and that will reduce the insect inoculum, pathogen inoculum, and also compresses the bloom into a shorter period. So your management practices would have more effectiveness. So it won't spread out. The bloom will not be spread out from like months. So it is a good underutilized practice, but it's a great potential tool. However, as I mentioned, due to the availability of alternatives like chemical control options, not many growers are implementing it. But now with the loss of chemical control options like chlorothalonils and mancozebs, there is a resurgence in interest, but the main drawback is lack of data. So as I said, this has potential to improve quality, and we can reduce inputs during the late water year and also the year after late water. But due to the availability of chemicals and lack of scientific data, not many growers are willing to use late water cultural practice. But due to the loss of some chemicals, there is resurgence in interest, and we are trying to understand various factors that contribute to successful late water year, so we can develop sustainable management strategies. For this project, late water project, which is funded by Northeastern SARE grant, here is our hypothesis. Critical criteria that impact the results or outcome of the late water floods can be identified, quantified, and evaluated. So by identifying various factors and quantifying them, we want to develop a late water decision-making model. Again, late water is recommended once in 3 years. So, but some years, if the growers implemented late water, they used to observe reduced yield, reduced production. So that's another thing that makes growers hesitant to make use of this tool. So now with this late water development tool, we hope to increase grower confidence and adoption of this cultural practice for sustainable cranberry production. So we started this project this year, which is a 3-year project, and we are planning to identify physiological, environmental, and climate factors that are associated with late water success. And once we identify the factors, we want to see which are most influential and how do late water and unflooded farms differ in yield, fruit rot, and fruit quality, and how to develop a late water decision-making model so that growers can understand which is the best year to hold late water and get the best out of it. And another project I initiated is improving cranberry fruit quality by understanding the microclimate of the bog. So we all know that the humidity, air circulation, cultural practices, all of these factors have impact on disease occurrence and severity, but that is not fully studied in cranberry. So we got funding from USDA NRCS grant, Conservation Innovation Grant. So we are studying 25 Massachusetts cranberry bogs. It's a 3-year project again. We'll be studying local microclimatic factors on these bogs, and also we'll also get the information from growers on cultural practices they have been implementing historically and for the next 3 years. And we'll also get weather data for these 25 sites. We installed weather stations already. So we'll understand what factors have influence on fruit quality and fruit rot. The goals of this project is to identify variables that are associated with fruit rot and what is the relative importance of each of these variables and their interrelationships. And the ultimate goal is to develop easy to adopt eco-friendly management guidelines. Excuse me. Guidelines. So, and long-term goals, which I haven't started this yet. Once we have microclimate data, and we would like to use that information for future prediction model. And right now we have a keeping quality forecast model, which has been issued since 1949 to understand whether it's a good time to hold late water or not, and how many fungicides are needed in a particular year, etc. But it was issued in 49. So, and this considered total sunshine hours, precipitation, and average temperature for March, May, April, and May. We don't know how relevant is this system to current cranberry production because there is change in climate, there is change in cultural practices. There are so many things that changed since 1949. So my goal is to look at this keeping quality forecast, either improvise it or revise it, or even develop a more practical forecast system for current cranberry cultivation. And I'm also interested in understanding the life cycles of fruit rot pathogens, which are not studied completely. So we initiated a study on overwintering sources of inoculum. Where are these pathogens hiding during the winter? Because when the cranberry goes into hibernation or dormancy, I don't want to use the word hibernation, but dormancy around November, where are these pathogens hiding? So we are studying that, and then how and when are they producing the inoculum to infect the next year's cropping cycle. So that is something we are studying in collaboration with USDA researchers in Wisconsin. So at present, there is no research literature on the sources of overwintering inoculum. We hypothesize that this pathogen survive in detached host structures or the debris left near the bogs. So we are studying 9 different bogs and trash piles, or the trash piles that are left close to the bog. Trash pile is nothing but all the plant material that is left on the bog during the water harvest or by natural fall on the ground. They're all taken away and the farmers leave them right next to the bog. So maybe that is serving as sources of inoculum. So we are studying those as well. And I'm also interested in microbiome studies. So we are going to get a grad student starting from this May. She's going to start some microbiome studies. So our goal is to understand microbiome of cranberry fruit with special focus on fruit. Because, every time we try to isolate cranberry fruit rot pathogens, we also get yeast and bacteria along with fungi, but we don't know what role they play in fruit rot. So we want to understand the role of cranberry microbiome, all kinds of pathogens. And we went to wild bogs. Now, wild bogs are just 2 hours from where we are. We have opportunity to study wild bogs in Massachusetts, in close to Provincetown, and like tip of the Massachusetts. They have some wild bogs which were never under cultivation. They might be hundreds of years old bogs. So we were getting berries from there and studying pathogens and beneficial microbes from wild and cultivated system. So never cultivated and cultivated. How does cultural practices change the microbiome? That is something we are interested in. And what factors contribute to these differences, if there are any? And what are the role of various microbes in fruit rot? As I mentioned, there is significant amount spent on fungi in fruit rot, but we don't know what the role bacteria and yeast that we have been isolating play in fruit rot. So we want to study all these with microbiome studies. So I hope I could cover this topic and make you understand the current cultivation in cranberry and fruit rot research. I'll be very happy to take any questions you might have. Once again, thank you so much, Shivo and Sujan, Liberty, and Soma for giving me this opportunity. Thank you. Thanks for attending this webinar.
Thank you. We have few questions we already collected, but we wanted to go for the interview first. Okay. And we have 4 or 5 questions, and after the interview, I will put it in the chat box, and you may answer as your wish. Shoma, I request Shoma to interview. It's not exactly the interview, but we are talking and wanted to know a little bit more for you, and many MSc, BSc, and PhD students are listening our webinar, and these interviews are really motivating for all of us.
Okay. So I'll be happy to.
Hello everyone. You are actually watching webinar 33 from Bioingene, and we are very fortunate that Dr. Leela, showed us such beautiful ways and the beauty and the importance of cranberry. We thank her for that, and we would love to know more about her. So my first question to you is that how did you build your career? Could you please share your journey as a plant scientist with our viewers?
Okay. Yeah, thank you. So it was a little complicated, but not so simple, but it's a beautiful journey. So I grew up in a village called Kormamidi, as I said before, and my family is all farming and business people. So we didn't have many people to seek advice when we wanted to get the career in plant science. But my father had this habit of, taking me around and ask people, their advice. So to best career path and what can she do and all that. So when I had interest in agriculture, when I joined in my undergrad, he took me to a bank manager and he suggested, why don't she try do writing GRE and TOEFL and then go to United States and get better exposure, of US education and research. That is the first seed. But then I didn't know how to pursue that. until I met Sujan. Sujan is my super senior in undergrad. So when I heard from him that he is studying for GRE and TOEFL, so I was asking him lots of questions, and he really, really helped me like a big brother. And he supported me, and he suggested, what to write and what to study. So that's how I started, learning more about it. But I did my master's in Kerala through ICR fellowship. That helped me too, because I was in sheltered Andhra Pradesh, my own world. But then going to Kerala first helped me a little bit with my language, because I studied in Telugu medium in Andhra. But then when I first went to Kerala, it was a little helpful because, that's the first time I was out of state where I had to communicate in English. There was no other option. So that was good. But then after that, I came to the US, and it was more of a worldwide experience I got after I came to the US. And then it was not a rosy, easy journey, because just 1 year after I came to the US, I had so many dreams and so many ideas, and I was still learning my project. And then 1 year after that, 1 year after I came, I was diagnosed with leukemia, a kind of blood cancer, in 2009.
Oh, my
so that was scary, because I was only 23 years old, and you hear this, in the midst of PhD. But that is when, when the hard times hit, that is when you know your strength. You would know how strong you are, right? So you are like, okay, well, I have two options, whether to quit or take it as a challenge and move forward. But of course, by God's grace, I got healed, and then I continued my PhD. And then, I think of course, hard times would come in different ways for people. But for my plant science career, I would say meeting the right people, asking the right questions at the right time, and willingness to learn and not give up. Those are very helpful. Sujan, as I said, he helped me understand the procedure for GRE, TOEFL, and that. So that's how, there are always people that are willing to help you, but if you ask them. So, keep asking the right question. Nothing is small. So just keep asking the right question and seek help, and then never give up. That's my big suggestion to any aspiring scientists.
Wow, that's great. What motivates you to work for the betterment of agriculture?
Yeah. So, what, I grew up in a rural background where farming is a major source of income. I have seen firsthand the hardships of farmers, how hard they work for very little they get. Their income is less, but passionately they go every day, like 5 o'clock in the morning, and then work whole day so hard. So that's the passion they have for farming. I have seen that. And I have also seen that there are smart people there who are smarter than me, but they don't have all the resources and the education that is required to achieve the dreams. For example, my father, he's one of the smartest people I know, and he has limited education. And that's why, his world became a little smaller compared to mine. So I thought, when he allowed me and gave me the best opportunities he could, whatever he could afford, I don't want to take them for granted. And by seeing the hardships of farmers firsthand, if I cannot help them, then, there is no purpose in pursuing this field. And then, but actually my passion and interest did not happen overnight. They developed over years, based on the situations I faced and the people I met. So it was like, of course, for anybody, passion, you would develop that passion, and you would understand your interest and your abilities and where you can help, how you can help through over time, And then now, as I look back, I think I'm more interested in safety of our farmers and health of our farmers, because, I myself faced leukemia. So now when I see farmers not wearing safe protective gear before they spray, I thought, that is something I want to educate farmers about, especially in India, that we don't use much protective gear for spraying. And I'm also interested in regulations in pesticide use and promoting sustainable integrated disease management. So these are the motivations I have for the betterment of agriculture. Not many people have opportunities what we have. So when we have them, use them for the best of our knowledge and to the utmost commitment we can give.
Right. According to you, which sectors of plant science research will prosper in the near future?
Wow, this is actually a really hard question because, I think every sector has its own potential for growth. And especially if you consider the technology that has developed in the last decades that opened avenues for revolutionizing agriculture in various sectors. So, more and more technology can be implemented now than before. Some of the technologies were not there before. Now that technology came into play, so you can utilize that and achieve more in any particular field. But if I have to think about some sectors, I would definitely say molecular tools because they definitely changed the diagnosis of diseases and also plant breeding efforts. We can diagnose even faster, and we can understand microbes in a soil or fruit much better with molecular tools. And also, it also reduced the time frame required for plant breeding efforts, because previously it used to be just traditional applied plant breeding requires decades of work before they can release a variety of hybrid. Now, molecular tools definitely came in handy that can reduce the time required for their efforts. That's awesome. So molecular tools is one area I would think would prosper in the near future. And also maybe drone technology, because drones are awesome in precision agriculture. And I think it will grow tremendously in the next decade or so because they reduce the time required for identifying disease. And also they are able to identify the pockets of field where a particular problem exists. So you don't have to spray the whole field or treat the whole field you can just particularly treat a particular area of the field. So you are reducing the required inputs while achieving the timely management. So I think drones are going to be really big in the next decade.
What are your views on the socio-economic conditions reflected on the crop domestication process?
I think, for any particular new crop domestication, it always comes down to demand, or the perception of the society for its need. So if a society sees the need for a particular crop domestication, of course, then it will grow. If the society doesn't see the need, and then, nothing else can matter. When I started my job at the Cranberry station in Massachusetts, to my surprise, some growers contacted me asking if they can grow rice in Massachusetts or in Northeast, because they knew that I worked in Texas A&M on rice crop. So they thought that she would be the one to ask this question. But I was surprised because only rice I know is cultivated in tropical areas like slightly warmer regions, Texas, Florida, California, Georgia, etc. But Massachusetts, where the we have long winters, short summers. It's like, how can you grow rice? But then, they are interested. So the farmers are interested. And there is a demand for locally grown rice here. So now I think short duration varieties, rice can be explored in Massachusetts. So as I mentioned, it all comes down to demand and perception of the need of the society. You see that, I'm sure they'll find ways to domesticate a crop.
What steps should be followed to inform people about the benefits of cranberry?
Yeah, it's a very interesting question. So, of course, illiteracy is still at high level in some areas in India. and around the world. So, but in general, more and more people are connected with the world now with the advent of internet and social media. And definitely there's more awareness on various topics now than ever before. And people are well informed. During my recent travel to India, I met a film director, Telugu film director. And then when I introduced myself as a cranberry pathologist, he talked to me about cranberries. I used to think nobody knows about cranberries in India. But then he was like, he had some health issue, especially urinary tract infections and stomach bug. And he was suggested by a friend to use cranberry cranberry juice on a regular basis. And he started consuming cranberry drinks and he was feeling better. So I think, word of mouth, advertisement has great impact, If a friend of us or if people we know, especially researcher like all of us, who has some facts and scientific data, if we tell somebody, definitely people will be more receptive of that information. We have to spread the facts, scientific facts about the cranberries or anything for that matter, whatever, any scientific data we have available that we think is useful for the society and people around us, we should keep informing them.
Could you give your views? Could you give your views or tips on time management that can be used while working in the field and in the laboratory?
Okay. Yeah, I'm not sure if I mastered my time management already, but I will explain what I do. Previously, I used to take so many projects because, I love to explore science and I want to do more and more. But then later I realized, especially in the last 2 years, I realized that prioritization is important. Taking like a couple of focused projects and managing them well is more important than taking 10 different projects and stress yourself and not being able to deliver the results, So prioritizing is really, really important time management practice. So now I am like, it's okay if I cannot do 10 different projects, maybe I'll take 2 at a time, or 3 at a time. So prioritizing is something I'm doing now that helps in my management. And also I'm trusting my brain less because previously I used to think that, I'll remember this fact. I don't have to write it down. But then now I'm trying to write everything down, record keeping either for planning or execution of your projects, lab or field level is important. Because my dad used to have a small sheet of paper every day and he will write down what he wants to do for that day. And then, he will cross them down when he is done. So that's something I liked because, if you write what you have to do, and then, then definitely you will manage your time well. Then, you think that, I will remember this, I'll remember that. So even for data management, because, our data is extremely important, because what we get out of our research would have great impact on farmers' lives and in agriculture and any crop data, or even for basic advancement of science. So record keeping is important, the details are important. So I would recommend, strong work ethic and remember the importance of data you have and, write that down, time management for best time management, writing things down and prioritizing and having focused research areas will help you be on track.
Yeah. That's great advice. What is your future aim in your field of research?
I have like several short-term and long-term goals. So maybe the immediate short-term goals are to identify novel methods of fruit rot management and develop true integrated disease management strategies. I don't want anybody to rely on, or any farmer to rely on one particular tool. Don't overly use chemicals. Don't overly rely on biologicals and maybe you end up less yield, So you just use a real integrated disease management strategies that will help you reduce inputs and improve quality. So my goal is to develop the integrated strategies. That's the first and foremost. And at this level in my career, I also hope to, reach out to more and more people and expand at various level. So one of my priorities is to find ways I can help Indian farmers, because I'm far from India, but I'm sure I'll find ways to help farmers in India and empower students as much as I can to achieve their goals. Because, I knew the hardship of not having people to seek advice initially, but then when I met Sujan and other my seniors who were willing to go the extra mile to help people in need, now I want to do the same. I want to help students, whoever are interested in pursuing higher studies and have questions on paths they can take, achieve their goals. So, and helping growers and helping students is something I have in mind. And for that, I hope to develop a social media and YouTube-based knowledge base. I don't know how far I'll reach, how soon, but I do have that in mind.
All the best for that. I'm sure you will do great. And finally, what would your words of advice be to the students and scholars who are new to the field of plant science research?
New to the field of plant science research, okay. Uh, it is funny, after my undergrad, I used to think that I would know everything at the end of the undergrad, because I studied 60 different subjects over a period of 4 years. So I'll have a lot of knowledge to answer any farmer's questions. But that did not happen. I came out of undergrad with more questions and I was like, oh, how do I recommend anything for farmers? Because they have been cultivating for decades and you only spend 4 years. So, but that knowledge I gained is different from what farmers gain. So I developed solid principles of science. So, and then again, my master's helped me to understand the methodology used in research, and PhD helped me to deepen my understanding of kind of research options you can do and be an independent researcher. So at every level, you would learn more, you will ask more questions, and of course, your dreams also will evolve. You keep working. Sometimes, the initial dreams you had may evolve to something else. Initially, my dream used to be to get a bank job and get married to a man who has a red Maruti car. That was my dream. But then now I don't have a bank job, and my husband doesn't have a Maruti
How
car.
old were you when you had this dream?
Maybe around 14, like job dream was an undergrad. Red Maruti car dream was 14. But now my husband is a plant breeder. He doesn't drive Maruti, he drives Nissan. So, dreams evolve for better. So it's okay, So just put your best foot forward and then keep dreaming and then never give up and ask questions. Never be afraid to ask questions. Never be afraid to say, I don't know. Because, that is another important thing I wanted to stress. When I came to the Cranberry station 2 years ago, as an assistant professor, I was like, I am the expert now. If a farmer asks me, I have to answer, that pressure I had. And then every time somebody asks me if I don't know, I used to stress myself out. I'm like, oh, I'm supposed to be the expert. But no, because every crop is different. Every disease is different. There is always learning curve. So if I'm able to say, I'm sorry, I don't know, but I learn, that relieves you of the stress. So now I'm like, that is a first step in learning. So now I'm like, ask more questions. If you don't know something, just say don't know, but you can learn. So that is the best advice I could give, just keep asking. Never be afraid to seek help too. If you are like, I want to do this research, but I don't know where to find tools. That's okay. You ask them, maybe 4 or 5 people you ask, one of them will definitely help you. So
that's great. Yes, you speak so beautifully and with so much passion, and also the way you speak about your dad, your father, it's beautiful. It's loved.
Thank you so
You
much.
must be a great, great person. So the questions have been provided in the chat. If you can, in the Zoom chat.
Yeah.
There are.
Okay. Chat. Okay. There are some questions. Let's see them. Okay. So SK Das.
Yes.
Is cranberry powder available in the market? What is the cost benefit ratio of cranberry cultivation per hectare? What are the market channels and preservation of cranberry in Indian condition? So I think cranberry powder is available in market. Because I have seen the products, but I have never purchased them. And then cost benefit ratio definitely depends on varies from one year to the other. And also how much you produce. But right now it's around like $30 a barrel. And they produce around 200 barrels per acre if it is a Stevens cultivar. And the cultivars differ too. Like, super hybrids even produce 300 barrels per acre. So the kind of cultivar you grow, and then current market price, and how much you spend on disease or weed or insect management, they all have impact on cost of cultivation. Some years it is more, some years it is less. And marketing channels, as I said, they produce lots of processed foods these days, because long back they used to rely on fresh fruit market, where they just sell fresh fruit, and which is used in consumption in Thanksgiving and also for other like around November and December, because they can only stay for 8 weeks at total. But now with processing, they are developing juice, concentrate, and other processed foods which are available at different markets. So there are more and more processed foods now, so of cranberries than ever before. And relevance of cranberry with Indian situation, I thought about it, but I don't know if we can grow cranberry in India because of the climate. Because cranberry around October in Massachusetts go into dormancy, and it requires around 3 to 4 months of chilling hours. I don't think, especially in South India, where we don't have that chilling temperatures, I don't think we can grow. That's my understanding for now. And how big is the cranberry industry? You can ask that. So right now, there are around 40,000 acres of cranberry cultivation in the US. And they are, I don't have exact number, but they have big impact on jobs in the US, and also with their processed food market. This is pretty big. And it's also, cranberries are also exported to other countries now. Again, more questions from Joshi and others on whether can it be grown in Indian climatic condition. Again, as I said, it requires chilling weather for dormancy. So I don't think so, but maybe if some people try, we would have better knowledge. Okay. So what are the conditions? Yeah. Please suggest IPM to control fruit rot in cranberry. Yeah. So it is, as I said, cultural control methods like late water, pruning, sanding, and chemical control. They all can be used to integrated disease management of fruit rot. Yeah. Okay, there is one last question by Manish Mandotra. If there is a drought-prone area, we can have a genetically modified crops. But in case mild weather or in drought area, there is a lot of rain. What would you suggest on opportunities in this field? I don't think I understood this question, Manish. But could you please write it to me at my email address? We can talk about it. I don't know. Yeah. Okay. Any other questions or? Yeah, and then can we reuse the flood water? Yes, I think we can reuse the flood water.
Okay. And one question I wanted to ask you that how much mechanizations is important, because in US there is a lot of mechanizations in the field to isolate the fruit, to wash the fruits, to pack and to harvest from field to directly to the plant.
Yeah.
But in developing country or in India, we are not using so much mechanizations technology. And so in this context, how we can cultivate such type of fruits, those are very little in the size, and they are not the long plants, they are like bushy plants, and they are not the good height that you can harvest. So what do you think that mechanizations will important for Cranberry cultivations?
Cranberry cultivation, as you mentioned, could be labor intensive. It requires a lot of labor. Till 1940s, they used to use the handheld scoops to harvest. But think about it, it's literally on the ground. You have to bend and do it. It's a lot of work. So mechanization, I'm sure, would have great impact in reducing the pain and also, the cost of cultivation too. Because, if you are like harvesting 1 acre, think about how many people you need to harvest that much in a day, because fruit will slowly degrade, right? It won't stay that fresh for so long. You have to do it in certain amount of time or certain period of days. So mechanization will help us achieve that in short period of time, so we can harvest. But it is labor intensive, requires a lot of people. And also in recent times, we have been reading about urbanization, and more young people are not considering farming as an option, career choice. So we have, moving forward, we will have less number of people in agriculture than before, but we have more need for food because of globally raising population. So it is going to be a challenge. We need more people in agriculture, and government has to find ways to attract young generation in agriculture, governments across the world. So mechanization may help the young generation, attract young generation to farming. Because if you are like, a lot of people see it as a lot of labor intensive, I don't want to do it. But if you are like using drones, it will attract young generation to agriculture. If you are using mechanization, they're like, oh, I want to do this. So mechanization would have multiple benefits: attracting young generation, reducing the time it requires, and also, doing any work efficiently in a timely manner.
And I wanted to ask you another question, is for just my curiosity, that when you use the sand to sanding the field, is there necessity to check the pH of the sand, because many times sands are very salty in such cases? So is it affect the plant, or you have some technique like flood it and wash out the something? What type of technique you have generally used in after the sanding?
Thank you, Shiva, for asking that question. I never thought about pH of a sand, because, in Massachusetts, when I first came in, when I heard about sanding, I was surprised, why would anybody put sanding? Why not some organic matter with high nutrients? But cranberry prefers sand, right? Because the bogs have more than 90% of sand. And then locally, we have around, in Massachusetts, we have 16,000 acres of cranberries. So most of them are located in southeastern Massachusetts. So maybe they all are of same sand, because they're all geographically at the same place. So I don't know if anybody really thought about differences in sand in this relatively small geographic region.
Okay. Yeah. So these are the questions related to your talk. And as I said that we got many other questions. Those are not exactly related, but as you say that you wanted to have students, some student named Manish Manhotra He asks that can you suggest a good institute, India and abroad? He had done his postgraduate MSc in genomics. So
Okay.
what will be the strategy to find out a good institute in India or abroad?
Okay, that's because I'm not in genomics. He's in genomics, right, Manish?
MSc in
yeah.
genomics.
Yeah, so there are several institutes that offer genomics, right? So my first suggestion is Google search what are the best genomics institutes, and then the professors. For example, if you say Cranberry Pathology in Google search, if you're interested in Cranberry Pathology, you will see like maybe 10 different scientists. You type on their profile and see what kind of research they are doing and contact the professor directly. Yeah, so like, oh, hi, Dr. Uppala, I'm Manish, I would like to study PhD. I would like to pursue PhD. Do you have any suggestions for me, or are you taking any students right now? So, because it's, as a professor, I need students to get my research done too. I need postdocs to get my research done too. So it's like, if I have some project which I need somebody I'm looking for, then I'll be like, okay, send your application. Similarly, for any institutes in India as well, you will even have better idea of what institutes in India offer genomics, right? Because, you are from India. So search and contact the professor directly. Maybe contact like 5 different professors in 5 different institutes based on what they are doing and whether you like that kind of research, and you will get an idea. But if you, as I said, if you need more information, please feel free to contact me. I shared my email address. I'll be able to help you or guide you in the right direction, at least.
Thank you. And I wanted to add something to your answer, because what I think that when you search someone, you just see that, okay, he did the genomics, and he will write something that, please take me, please, I wanted to join your lab. But it's not the right process, and it's not the right way to take someone that you find your email address and email anybody. So I suggest this is one of the reasons that when we are bioengine, starting the bioengine, because we already had this type of timing and scenario after BSc, where we should go for the dissertation, after MSc, end of MSc, where we did the dissertation process, then find a lab for the PhD, then find a lab for postdoc. So it's a cycle, and everybody do that and find something. So I suggest, Manish, first you choose some field, then go for the particular field, go for a few reviews, go for a few research papers. Sometimes research paper is very tough, but reviews are, you can go for the review. Then you design a plan that which papers are fitted on in, because it's not possible that everybody doing the same work or your interest fits with others. So find out your intention, your interest, because you already did the MSc. Then you should start the searching, and I'm sure like in bioengine, there are a lot of speakers. Those all have the experience on that. You can start with these speakers also, because their talks are available, their research works are available. And you obviously it's their profile are available in Google Scholar, and its org ID is available in the online. So go for that. Go little bit their studies. They at least their 5 years work. Start with the 1919 or 2018 or 19, then go for the up to 2021. Then you can realize that, okay, he is doing that, because sometimes they are not doing their work as they did in their PhD. So it's sometimes different. Sometimes somebody is working on zoology, but now he is working on genomics. Sometimes some people are working on biochemistry, but now they're doing the agriculture. So it's better to search not by their degrees, but their recent work. And before that, go for a few review papers. Otherwise, you can just email somebody, because why I ask you, because I'm not such personality that I got email, but I can understand this thing. Many people are email as me, where is my certificate? But that is. But which webinar, in which context, which date? Because there are a lot of webinars, and how I know that you are asking for which certificate? And sometimes they are not going for the details, because already many details are available on the institutional website. They are specifically mentioned which type of openings are there, which type of research are there, what will be the GRE score they needed, and what is the procedure. And sometimes they give so many funding opportunity to go to. Before I'm wanted to say, before disturbing any scientist, first go for these details. If you fit in that way, you ask the direct questions, specific questions, I'm sure you're getting the mail back, answer your email. Otherwise, I don't expect to email back from a reputed scientist, because they have so many work to do, and it's not their main work. So I know it's a little bit rough and harsh, but it's a fact. It's a fact. It's everybody knows this can happen, and try to email something. Try to focus on choosing your subject line. Sometimes we are just delete the email, seeing the subject line. So it's a trick. You need to learn this trick, and obviously Bioingene try to help this building, like how to write, how to communicate, but we are not doing anything about the how to write, to how to catch a scientist for your purposes. But there are a lot of openings. There are a lot of openings here, and we also have a dedicated tab. This is called Bioingene Careers, where you can find so many openings on PhD, postdoc, and research. Okay.
So I have one thing to add. So I agree, most of the researchers would like to advertise the positions, but not all of them. So that's why I would, if you, Manish, one thing is, sometimes you would know what kind of research you want to do based on your experience before. Sometimes you want to expand your skills in PhD. I'm like, I did integrated disease management in master's, but now I want to do epidemiology kind of research in PhD. That's okay too. You can contact a professor saying that, so far I only did this, but I want to expand my skills in epidemiology, or I expand in molecular plant pathology. And I saw your profile and you are doing this area. I'm a quick learner. I'm enthusiastic, just say what about yourself, and then it's okay, because that's the opportunity. PhD or postdoc or masters, these are the times you will build your skills. So this is not the time to stick with one thing, if you're not interested in that, just for the sake of continuing what you started. If you are like, okay, I studied this, but I'm more interested in learning my tools, more tools, that's okay, contact them. But as Shua said, most of the researchers are busy. Try to focus on the details, specific messages. But if some information is already there in the website, like GRE score, you already have it in the website, what they accept, don't ask that question, because that's already there. But if there's information is not there, ask them. Most of the people would like to reply, but if their time doesn't permit, that's okay. You keep trying it, and then one day you will get what you wanted. But not all the professors advertise, but as Shiva said, that's a great way to start. If for advertised position, definitely there is a position. But on the other hand, if you keep writing to people, then sometimes they might have, sometimes they don't have. So keep doing your research and keep learning. If you want to expand your toolbox to more than what you did, that's okay too. Just learn.
Thank you, Leela. It's the end of our today's webinar session. So thanks for participating in this